Welcome everyone. Let's discuss another question from GATE 2023 Biotech exam. The question says the number of different possible ways of forming 5 intramolecular disulfide bonds with 10 cysteine residues of a protein. Cysteine is a sulfur-containing amino acid which carries thiol group in its side chain. So this is the thiol group, and two of the cysteine molecule can form a disulfide bond under oxidizing conditions to form this disulfide covalent interaction. Now, this disulfide bond formation can take place within the same polypeptide chain, and this I'm calling it as a intramolecular, or it can take place between the two chains, which I'm calling it as a intermolecular. So remember, intramolecular within the same polypeptide chain, while the intermolecular is between the two chains. All these interactions, be it intramolecular or intermolecular, these helps in stabilizing the tertiary and quaternary structure of the protein. So now let's calculate the total number of disulfide bond formation for this question. So let's assume that this is a polypeptide chain and there are total 10 cysteine residues. 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. Let's give these cysteine residues a number. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We are considering a single polypeptide chain because it has been asked in the question for the intramolecular disulfide bond. Now, let's consider the first cysteine residues. What are the different possibilities for this cysteine to form a disulfide bond? So the first cysteine can form a disulfide bond with either 2 or 3 or 4, 5, 6, 7, 8, 9, 10. Total number of possibilities are 9. Now let's assume this first cysteine is forming a disulfide bond with a third one. So the first and third cysteine residues are already involved in a disulfide bond formation. And for our further calculation, we are not going to consider the first and third cysteine residues. Now, what are the different possibilities for the second cysteine residue to form a disulfide bond? So the second cysteine residue can form a disulfide bond with either 4 or 5 or 6 or 7 or 8 or 9 or 10. So the total remaining possibilities are 1, 2, 3, 4, 5, 6, 7 for the second cysteine residue. Again, let's assume the second is forming a intramolecular disulfide bond with a fourth cysteine residues. So now 1, 2, 3, 4 are already involved in the disulfide bond formation. Now, what is the possibility for the fifth cysteine residues? And now it can form a disulfide bond either with 6, 7, 8, 9, or 10. So the total possibility for the fifth cysteine residue is it can form a disulfide bond with either 6, 7, 8, 9, or 10, which is 1, 2, 3, 4, 5. Similarly, let's say this fifth is forming a disulfide bond with a sixth one. So for the remaining cysteine residues, what are the different possibilities for forming the disulfide bonds? It's going to be for the seventh, it's going to be 3. And for the remaining 2, it's going to be, for example, for the ninth last residue, it is going to be 1 because only 1 is left. Now, these are the total number of possible ways of forming a 5 intramolecular disulfide bonds with 10 cysteine residues. Remember, a single disulfide bond involves the involvement of 2 cysteine residues. So if there are 10, there are going to be total 5 intramolecular disulfide bonds. If there are total 8 cysteine residues, there are going to be total 4 intramolecular disulfide bonds. The numbers are going to be different for the intermolecular. So this number corresponds to 945. So coming back to this question, the number of different possible ways of forming 5 intramolecular disulfide bonds with 10 cysteine residues of a protein is 945. As I said, the numbers are going to be different for the intermolecular. What I would recommend you to try the same question with the intermolecular disulfide bonds and report your answer in the comment section. I hope this solution is clear to each and every one of you. If you do have any concern, please let me know. Thanks for watching the video.